I'm Dr. Tom Lane, the Chief Science Officer at Immunis Incorporated.
I'm Dr. Nicole Birchtold, Principal Scientist at Immunis.
I'm Dr. Scott Greilich, a Research Scientist at Immunis.
Our cells secrete factors that influence the immune system and prevent age and disease-related immune dysfunction. These secreted factors are collectively referred to as the secretome. As we age, we experience a gradual decline in our immune function and a decrease in these beneficial cellular secretions. Consequently, we become more vulnerable to degenerative illnesses, including muscle atrophy.
Muscle loss, or sarcopenia, is a major contributor of immobility in elderly adults and greatly reduces quality of life. There are currently no treatments available to attenuate muscle loss or to enhance muscle regeneration.
Immunis has developed an investigational secretome from clinically compliant human cells in hopes of addressing the unmet clinical need associated with progressive muscle loss. Our collaborator at the University of Utah, Professor Micah Drummond, spearheaded the preliminary experiments using Immunis' investigational secretome in aged mouse models. The goal was to examine the efficacy of the secretome on muscle mass and function during periods of disuse and recovery. These data were published in GeroScience, and today we will be discussing the results from these studies. In an effort to understand how the aging process affects muscle mass and strength, a team of scientists conducted a groundbreaking experiment. 3 experimental conditions were tested using 22-month-old mice, which are equivalent to humans in their 60s. The study design was broken down into 3 parts. First, investigators tested whether intramuscular injections of the investigational secretome product, administered every other day for 14 days, affected the muscle of mice with full mobility. This is referred to as the ambulatory control. The second condition tested whether administering the secretome every other day for 14 days affected the muscle in mice that could not use their hind limbs. This is called hind limb unloading, and it simulates muscle atrophy caused by disuse. The final condition tested whether 14 days of hind limb unloading followed by recovery period with secretome injections every other day for 7 days affected the muscle. Hind limb muscle mass and strength were measured for each condition, and the results were compared to PBS-treated controls. For ambulatory mice, the secretome significantly improved muscle strength, muscle mass, and muscle fiber cross-sectional area when compared to PBS-treated controls. The secretome treatment showed greater BCHP, less collagen IV, demonstrating improved collagen turnover compared to PBS controls. In conclusion, these data suggest that the investigational secretome enhances muscle size, improves muscle function, and increases collagen turnover in aged mice.
The Drummond lab next investigated whether the secretome induced recruitment of macrophages, which are immune cells that can influence muscle health. Immunofluorescent imaging of the muscle showed a strong increase in macrophage markers, including CD68, CD163, and CD11B. This increase correlated with a nearly 3-fold increase in the number of PAX7 positive cells, which marks for muscle stem cells known as satellite cells.
In conclusion, these data demonstrate that our investigational secretome enhances macrophage and satellite cell numbers in the muscle, suggesting the secretome may function through an immunomodulatory mechanism. In other words, there is strong evidence that the investigational secretome is playing a key role in the immune system and muscle health. The next goal was to determine whether the secretome had an effect on muscle during periods of disuse in aged animals. Under conditions of hind limb unloading, secretome treatment prevented a loss of grip strength at 7 and 13 days compared to control mice. The investigational secretome product also increased muscle mass and cross-sectional area when compared to PBS treatment. In addition, secretome treatment condition showed higher BCHP and collagen IV levels compared to PBS controls, indicating improved collagen turnover. These findings suggest that the secretome may be effective in mitigating disuse-induced muscle atrophy and fibrosis in aged mice. The next goal was to determine whether the secretome treatment also recruited macrophages and increased satellite cell numbers in mice during hind limb unloading, a model of muscle disuse atrophy. The secretome increased the number of CD11B positive macrophages and muscle stem cells compared to PBS treatment. During hind limb unloading, secretome-treated mice have more macrophages and satellite cells in muscle than PBS-treated mice. They next determined the effects of the secretome administration on muscle in aged mice following a 7-day recovery period after hind limb unloading. Secretome treatment resulted in greater grip strength, muscle mass, and increased fibrous cross-sectional area compared to PBS-treated controls. Also, the investigational secretome increased the BCHP to collagen 4 ratio as previously observed. In conclusion, the secretome improved soleus muscle mass, promoted muscle growth, and decreased the amount of stiff collagen in the muscle during recovery in aged mice following disuse atrophy. The last goal was to examine the effect of secretome treatment on muscle macrophages and satellite cell numbers following disuse. As previously observed, the secretome resulted in more CD68 positive and CD11b positive macrophages and a greater number of muscle satellite stem cells. In conclusion, these findings suggest that secretome treatment may promote muscle recovery from disuse in aged mice by increasing macrophage and satellite cell numbers, which are both essential in the regeneration process. One of the measures of muscle regeneration is myonuclear fusion. Myonuclear fusion occurs when satellite cells fuse with existing muscle fibers, resulting in a single muscle fiber with multiple nuclei. It is hypothesized that the additional nuclei provide growth and repair factors for the muscle. The study found that when mouse muscle cells were treated in vitro with secretome, the percentage of cells with myonuclear fusion significantly increased, and myotube growth also increased. This research suggests that the multiple nuclei provided by myonuclear fusion may promote additional growth and repair benefits for muscle cells. In aged mouse models of muscle disuse and recovery, the secretome improves muscle mass, volume, and function, increases satellite cell numbers in the muscle, suggesting that it promotes muscle regeneration and growth, recruits macrophages to the muscle, which is necessary to promote muscle tissue remodeling and muscle regeneration, increases collagen turnover, thereby reducing fibrosis, which is more conducive to muscle regeneration. The investigational secretome also exhibits muscle-specific effects by promoting myonuclear fusion and myotube growth in vitro. The results clearly indicate that the investigational secretome has a beneficial effect on ameliorating the severity of age-related muscle atrophy, and this has led to an ongoing FDA-approved clinical trial. In addition, at Immunis, we're conducting further experiments evaluating the effects of the secretome in improving other age-related illness or degenerative diseases.